So there's a group on Facebook called Buy Nothing, Sell Nothing, something like that. Basically, it's a group of people who just give stuff away, and also if they need stuff, they ask for it. It's supposed to be like better for the environment because you're not, throwing away stuff that somebody else might use. Anyway, I found a guy in there who lives in Nitro. It's on my way to the recycling place, and he's been throwing away all of his cans. And he's like, "Oh, I'll give you my cans." So I just swing by his house on the way. It's nice. It's only one person, but every little bit helps. Good morning, Hill.
Good morning.
And O'Brien, do you need help with the Punnet square?
Yes.
That's good. Yes. We're recording, so other people can learn from this. All right. So what was your genotype?
I genotype what?
That would be, I think, on the
little h.
Okay, so you were little h. And what was your hypothetical mate's genotype?
Big H, little h.
Okay. So when you go through meiosis and you produce sperm, your alleles are going to split. But anyway, you only have little h, so no matter what, you're going to get a little h, a sperm with one little h in it, no matter what. This person, their egg, when they go through meiosis, their big H is going to split from the little h, so 50% of their eggs are going to have a big H in them, 50% of them are going to have a small h. So now we look at it. This square right here, we bring it together, the big H and the little h. Here we have a little h coming together with a little h. Here we have a big H coming together with a little h. And here we have a little h coming together with a little h. So as far as genotypes are concerned, I see two of them. See these two, and I see these two.
So the genotype would be 2.
Exactly, 2 to 2. And some people reduce that like you would a fraction. So I'm only saying this because I'm recording. So if you write 1 to 1, you're not incorrect. But yeah, technically it is 2 to 2, right? You got 2 heterozygotes and 2 homozygous recessive.
And the phenotype would be the same thing, right?
Exactly, yep. Because you have 2 that are expressing the dominant trait and 2 that are expressing the recessive trait. Good job. Yeah, that's it. I will erase that though, because anybody who comes in online, they're like, "Wait, are we talking about this again?" So, not to fear.
I just shared it with you, and I fixed some of the stuff.
Great. Let's see. Delete that. That's good. Yep, you fixed all your mistakes, so you are done with that lab. Nothing more to fix. And your grade is updated. All right, let's see. I wasn't paying attention. Let's just start from the top here. Cooper's here. Breeden? Not yet. Still have a couple minutes. Elias? Not yet. Davis? Not yet. Lloyd is online. Hamrick? Not yet. O'Brien is here. Ray is online. Baumgartner, Michael, not here yet. Oh, yeah, he is. I heard you ding. I assume that's what the ding was for. If not, just let me know.
Yes, sir.
Okay. Pena, not yet. Sigmund, not yet. Bayard, not yet. Johnson, not yet. Huffman, not yet. Hamlet, not yet.
Huffman is here.
Oh, thank you.
Thank
Good
you.
day. All right, Fitzpatrick, not yet. Thornabar is here. Oops, fix this. Lacey, not yet. Tyrus is here. No Burn. Hill is here. And Sullivan is not here. So have I missed anybody? I'm missing a large majority of the class. That's not good. Oh well, let's slowly get started. I say slowly. Maybe people partied too hard last night and they're just slow to get up. So, as a reminder, independent work is basically due in a week. It's not this Monday, but the following Monday. Some of you haven't done a thing yet, haven't written a single paper. So I'm gonna go ahead and tell you the common mistakes that I see because I'm recording this, so this will be great. So when you ask for leniency or say, hey, I didn't know, I'm gonna send you a link to this video. The two common mistakes I see at the end of independent work, they all boil down to the same thing, which is that you didn't understand how I grade. And the reason you don't understand how I grade is because you haven't turned anything in yet. You haven't turned in any written work. The biggest thing when it comes to not understanding how I grade is the biggest mistake is people assume that everything they turn in is worth 10 points. Because I have said, and if you don't pay attention, which a lot of you don't, you might think, oh, every time I turn in a paper, it's worth 10 points. He said a paper is worth 10 points. And I have not. What I've said is a full page is worth 10 points. So that's the first common mistake I see is people on the Sunday night before this is due on Monday. So on the 24th, they're going to send me 10 papers and then I'm like, whew, got it. And then on Monday, I'm going to say, nope, that's not 10. That's not 100 points. And then you're going to get a low grade and you're going to be disappointed. A similar mistake I see people make instead of sending me 10 different papers, they have it in their mind, and this is a little bit more accurate, that each page is worth 10 points. And that is correct if it's perfectly written. So for those of you who have turned in papers, you know I deduct things. And I don't count this and I don't count that, and I squish paragraphs together. And if you've done papers, you know what I'm talking about. So that'll be another mistake. Someone will turn something in on Sunday night, and it's going to be what they think is 10 pages. And then it's going to be like, and then they'll find out it really was not 10 pages and it wasn't 100 points, and they're going to be upset. And then I'm going to send them a link to this video. Another common mistake, and some of you have already done it. Thank goodness. And I'm glad you guys have made these mistakes for those of you who have done it, because that's how you learn from it. You cannot reword an article. So don't go in there and I've given you examples of this, like on the Google Classroom, but you can't copy something and paste it and then just change a few words. That's not writing. That's, that is essentially, wait, I'll take that. Sorry. That's all right. I'm at the close the door anyway. I'm putting out noise pollution. Thank you. So yeah, make sure, and this is why I've been begging you for the longest time. I have to keep saying, hey, at least write one small paper. Answer one question about biology so you can get a feel for what it's like to turn something in. You get a feel for how I grade, and you can understand that, what plagiarism is. So please start now. Please don't wait until midnight, right? Because on Monday, on October 25th, at 12:01 a.m., it's too late, right? You have until midnight, Sunday going into Monday of next Monday. So please, please, please get to work. And you're not in this alone. I love it. One of you's already sent me an email and said, hey, can you give me some ideas? And that's, what prompted me to send out that announcement last week where I was like, hey, here's a list of questions you can answer for independent work. So please get it done. And please remember, they don't have to be long papers. You can just answer a question. Why do farts smell so bad? According to buttmagic.com, Actually, you'd probably lose points for something using the source like that. But yeah, just, think of a question, answer it, include your source. That's it. Please, please, please. Because you're running out of time. And I've got that on record, so let's move forward. Does anybody have questions about independent work while I pull up where we left off on the slideshow? Okay. So, and because independent work is so important and so upcoming, so to speak, next week's lab is going to be online. I hope to share it with you today or tomorrow. And just like last time we had an online lab, as long as you do it before lab, that day, then you don't have to meet me online. And if we do meet online, all I'm going to do is sit there and, it's going to be a really simple lab. But anyway, I'll be there online for you to answer any questions you have, and you'll just need to show up just for attendance. Again, assuming you haven't already done the lab. But I'll send that out today.
So you're gonna post the lab
Today?
Yeah,
either today or tomorrow. Yep.
Until next Wednesday to do it, or next Tuesday.
Exactly. Yeah. All right. Mitosis and meiosis. We already talked about that. Here we go. The origins of genetic variation. Now this is important. This is important for more than just this chapter. So soon we're going to get into evolution, and to understand evolution, you need to understand genetic variation, because without genetic variation, there would be no evolution. So this is a very important part of this chapter regarding the entire semester, not just this chapter. And we've already talked about the origins of genetic variation. Whoops. I just didn't put it in those words. I didn't say, all right, these are the, hold on, I see a question. 1 second. I see a hand was raised. Did you have a question?
Oh, I was just going to tell you that Barry's network connection wasn't working and kicked him out, but he back.
Oh, great. Thank you. Good looking out. Anyway, yeah, so the origins of genetic variation. We've already discussed these things, at least one of these things, when we talked about meiosis. So there's one more thing we're going to talk about. And again, this will be important moving forward. So pay close attention. This first slide is more like an introduction to what we're about to talk about. And it points out, your book points out, that the offspring of sexual reproduction are genetically different from the parents and from each other. You already know that. So you can write it down if you want. But I assume if you're a note taker, you've already written this down, because I've already said that before. Because you know that the products of meiosis are 4 genetically unique cells, right? So that's just the gametes, right? That's just the sperm or the egg. And then you put those things together, obviously it's going to be even more unique, because you have one unique thing meeting with a separate unique thing. It's even more unique. So you already know this. And if you need a little cheat sheet on how to remember that, picture yourself, then close your eyes and picture your parents, right? Even if you look a lot like one of them, you're obviously not a clone of one of your parents. You are genetically unique from your parents and from your offspring, or excuse me, your siblings, if you have any. So the question is then, how do we get this genetic variation through meiosis? And I've already given you one of the answers, like one of the ways this has happened. And I'll give you a hint, because I messed this up. This happens in prophase of meiosis, prophase 1 of meiosis. And I accidentally, when I described it as happening in metaphase, because it's easier to picture that way. But it actually happens in prophase. So maybe all those hints will remind you. Anybody remember? Actually, we'll come back to that one. I'm sorry. We'll come back to that. So you go ahead and marinate on that, and we'll come back to it. Maybe you'll have the answer by the time we get to it. So again, this slide here is just an introduction. Nothing here will be on the exam other than the first bullet point, but I've already given you that, right? If it's sexual reproduction, then the offspring are unique. Any questions about that? All right, here's the first one. And this first one, which is the independent assortment of chromosomes, this is a little bit complicated. It's very important, and we'll talk about it again in upcoming chapters. What this is saying, and there's a lot of words on that slide, but basically all we're saying here is that you know all the homologous chromosomes meet up in the middle, right? For meiosis 1. We already know that. And I'll draw it right here. You know, if we have a cell, and then we're in metaphase of meiosis 1, then we're going to have all these homologous chromosomes lined up, right? Of course, we have 23 of them lined up, so this is a much easier version. They're all lined up in the middle. They've got the mitotic spindle attached. There's a tug of war going on because we're trying to separate those homologous chromosomes. Now, in this case, let's just say the mom equals, the red are the chromosomes you got from your mom, and the black is chromosomes you got from your dad. Pretty simple, right? And there we go. We're looking in the middle of one of your cells, not yours, because again, you would have 23 of them. We have 1, 2, 3, 4, 5, 6. This is some creature that has 6 pairs of chromosomes, whatever. And the chromosomes in the red are the chromosomes they got from their mom. The chromosomes in the black are the ones they got from the dad. Does this picture make sense to you? Because I know we haven't talked about this since Wednesday. So I want to make sure that you guys understand what I'm saying when I talk about metaphase and the homologous chromosomes are lined up in the middle. Everybody sees that, right? Okay. All we're saying with independent assortment is, notice that it's not like, whoops, I'll do this. There we go. It's not like all the black ones line up on one side and the red ones line up on the other, right? Or it's not like there's a pattern. Okay, good, there's not. It's not like it's red, 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 right? All independent, all this is saying, independent assortment, is that when they line up in the middle, there's no rhyme or reason to how it happens. So it's not all mom's chromosomes on one side and all dad's chromosomes on the other. It's not always going to be 50-50, right? There is a chance that you might have a sperm cell that has 100% of your dad's chromosomes or 100% of your mom's chromosomes. Or it might be 75% dad chromosomes, 25% mom chromosomes, right? There's just it could happen anyway. So that's what independent assortment is. That's the most important concept that you need to understand, because without understanding the concept and understanding those numbers don't make much sense. And I'm about to talk about those numbers, but let me move this so you guys can see what I'm talking about. I don't know. No one spoke up. Hope maybe you could see it, maybe you couldn't. But this is what I'm talking about. See how I've drawn these chromosomes? There's no rhyme or reason, right? Sometimes the red ones are on the left, sometimes the black ones are on the left. That's what I'm saying. The mom and the dads, the way they line up, it's completely different. And that's for every time this happens, it's completely different. So it's not like whatever creature I'm looking at, it's not like 100% of their sperm are going to have this arrangement. No, they're going to have any kind of arrangement, because every time it goes through meiosis, this happens independently. That's all independent of each other. Yeah, I think there's a better word than independent assortment, a word that would be better to help you remember it, but I'm not going to give you that because that word's actually used for something else, and we'll talk about that a little bit later. So you might be thinking, why do we call it independent assortment? And you'll understand why it's called independent.
I have a question.
Yes.
Okay, so I'm a twin. Would ours look the same? Like, would ours be the same?
No.
Okay.
Because so when you're going through meiosis, you start with a normal cell. So you start with the same material, right? Because you have, if you're maternal twins, if you're identical twins, then you have the same chromosomes, right? So you have, if you got chromosome number 2 from your mom, how do I put this? You guys have the same chromosomes. But when you go through meiosis, remember that whole purpose is to cut in half your genetic information. So the way it does it is random. So not only, that's such a good question. Not only are your eggs different than your twins' eggs, but your eggs are different from the other eggs. Each egg you have should be unique because there's so many different ways that they could line up. Mom on this side, dad on that side. But you guys can see the picture, right?
Yes, we can. Thank you.
Okay, you're welcome. That's a great question. And that's what brings us to this part right here. For humans, this is where we actually give it a number. Because humans have 23 pairs of chromosomes, that means if I were to ask you for an independent work assignment, you would kill me for this because you wouldn't get it done. No one would, I don't think. If I were to ask you, like, all right, I want you to draw this. But instead of 6 pairs of chromosomes like I've drawn here, draw all 23. Now I want you to draw every combination of how, mom on this side, dad on that side. If you were to do that, there would be 8 million possible combinations. So what that means statistically is if you were to look at a sperm sample, for example, and if you were to look at 8 million of your sperm and look at how the chromosomes are lined up in the middle, statistically, you should expect that each one of those would be unique. It's the basic way to put it. So there's some rules of probability that we'll talk about a little bit later. For now, just suffice it to say, if your sperm have 8 million, when you make sperm or eggs, you have 8 million possibilities, right? You have 8 million variations of sperm or egg or gametes that you can make. Same thing with your partner. So if you take like the one, here, let me back this up. If we're talking about you are the product of that one sperm and that one egg from your parents, right? Now, that one sperm from your dad, that was a 1 in 8 million sperm, right? You could have been any of 8 million or 799,999 other possible variations. Same thing with your mom, right? That's just 1 out of 8 million different types of eggs she has. So then you combine that, the fact that you're 1 in 8 million there and you're 1 in 8 million there, that means you are 1 in 64 trillion as far as simply which egg and which sperm came together. That's how unique you are. So if your parents really like doing it, if they were to have 64 trillion kids in their lifetime, the chances are you would still be genetically unique. And this is just talking about how these are aligned. There's even another part of the equation that we're about to throw in that makes it even more unique. But yeah, just based off of the way your mom and dad chromosomes line up, that's how unique you are. I guess the question here is, do I require you to know that number for the exam? And I'm not sure. So for now, I'll just go ahead and study it. I'm thinking for this exam, I might have extra credit. And I think if I do, then something that specific would be on extra credit. Like, how many possible combinations are there for your gametes? The answer is 8 million. How many possible combinations are there for a sperm and an egg fertilizing? 64 trillion. And that's a big number. I saw a meme. I'll share the meme with you later. Never mind. All right. Any questions about this? All right. Forget my stupid drawing. Let's look at the drawing from your textbook. Your book really does a great job because in this example, this critter, this organism, this species, whatever, it only has 2 pairs of chromosomes, unlike our 23. So it's really easy to picture, right? So with this thing, there's only 2 possibilities. You can either have all mom on one side and all dad on the other, or not, right? That's it. And of course, it's going to be a lot more complicated when you have 23 of these as opposed to just 2. But yeah, that's how that works. So anyway, let's look at possibility number 1, where you have all mom on one side and all dad on the other. You go through meiosis 1, which means the homologous chromosomes have split, at which point you're only left with the sister chromatids. And then those go through meiosis 2, which splits the sister chromatids. And now here you have 2 cells that have all mom chromosomes and 2 cells that have all dad chromosomes. And then this other possibility, again, you go through meiosis 1, so the homologous chromosomes split. So now you only have sister chromatids, but now you have them from both parents. And then those things split. And again, now you have each one of those 4 from each parent. And again, this is a very simplified version. This is something with 2 pairs of chromosomes as opposed to 23. So this picture is just an example. Nothing to study, nothing to memorize. This is just trying to show you what I've been trying to tell you about how chromosomes independently line up in metaphase. There is no rule to it. So I'm definitely not going to ask you any questions about this. You don't have to memorize. Like, I won't say, if an organism has 2 pairs of chromosomes and all the mom and dad chromosomes line up on each side of the cell. Nothing like that. Nothing to memorize here. Any questions about this? All right. I will point that. Nah, I don't want to make it any more complicated. All right. So that is one type of genetic variation. And obviously that's very powerful, right? Because like I said, 1 in 64 trillion is what you are. That's a very unique thing. But yet it gets even more unique. And this is the one we've already talked about on Wednesday. Does anybody remember the unique thing we talked about from prophase of meiosis 1? And think about chromosomes too. Think about this, where I say, oh yeah, mom chromosomes are lined up this way, dad chromosomes are lined up that way. What is something, looking at this picture, that can make things even more unique? And I've already said it. And it is one of the things that defines meiosis over mitosis. So you do need to know this. This is not trivial. I don't blame you for not knowing it right now because I only taught it to you on Wednesday. You haven't had much time to study it.
Sexual reproduction.
Sexual reproduction, we have talked about that, and that is what we're all talking about. So you're on the right track.
Is it when they exchange?
What is it?
Is it when they exchange segments?
Yes, exactly. So for example, here, we already talked about, again, imagine this was 23 pairs, 64 or 8 million combinations or whatever, 64 trillion combinations of you getting fertilized. But imagine too that not only do we have all this different variation of how they line up, but also imagine now we're swapping stuff. I know this might be hard for you to see in person or online, but I give you a picture from the book. So now we still have all those different genetic possibilities, plus though in the fact that it's not completely mom chromosomes or completely dad chromosomes. We have something called crossing over, which, as Baumgartner said, was the exchange of corresponding segments between homologous chromosomes. And you already know that. We've already talked about this. That's when they get close to each other and chunks of the dad chromosome and chunks of the mom chromosome, they swap. So then you have a dad chromosome with mom stuff on it and a mom chromosome with dad stuff on it. As we already said, and as you need to know for the exam, it occurs in prophase 1. I'm going to underline this. As far as I know, you're the first semester I've made this mistake, but the important part there is that it happens in the first meiosis. Again, yes, it does happen in prophase, but the first time I talked about it, I drew it in metaphase, and I'm still kicking myself for that. But anyway, yeah, it happens during the first prophase, or the first meiosis. And the way you can remember that is, remember, the whole purpose, the whole thing that happens in meiosis 1 is that these homologous chromosomes are separated. So if you can remember that, then you would have to remember that this crossing over cannot happen in meiosis 2 because the homologous chromosomes are no longer in the same cell. So they cannot cross over. They're not together anymore. So again, this slide is a refresher. We've already said this before. This is not new information. It's just given to you again. But are there any questions on what's on this slide? The next slide I'm going to show you is a picture of what we're talking about. And it's going to be an even more simple version. So instead of having 23 pairs like humans have, or instead of having 6 pairs like this random thing I drew, or instead of having 2 pairs like the last picture we saw, this one's only going to have 1. Here, this thing only has 1 pair of chromosomes, 1 from its mom, 1 from its dad. You can see here, they're getting a little fresh with each other. They're rubbing up on each other. What happens? They swap segments. And I remember, I don't know if you remember, but the first time I showed you a picture of this from the textbook, they only swapped 1 portion. It was like 1 little portion of the leg. Here, they swapped 2 portions. And like I said in that lecture, when they only showed the 1 portion, I said sometimes it's 2 portions. Sometimes it's 1. Sometimes it's none. Sometimes it's little chunks. Sometimes it's big chunks. So this is so completely random. So again, think about the 64 trillion different possibilities of chromosomes you could have gotten. And then add to the fact that those aren't even the full chromosomes. You might get, yeah, those 64 trillion different combinations of chromosomes, but then what? Is it going to be a fully mom chromosome, a fully dad chromosome? Is it going to be this much mom, that much dad? It's really complicated. So are there any questions, excuse me, about crossing over? All right. We're almost done with this chapter. Excuse me. Let's talk about when something goes wrong. And we're specifically going to talk about when meiosis goes wrong. And before we do that, I want to point some stuff out that will not be on the exam, that I feel like it's important. And I don't think your book covered it. If they did, then that means I accidentally skipped it in the PowerPoint. Someone let me know if I did. I'll go back and fix it. And I'll give you a little extra credit. But If you remember, when we were talking about mitosis, there was a little subcategory there where we talked about cancer, right? And I said cancer, it was basically where your cell cycle is messed up, right? Because you're not supposed to be constantly going through mitosis and making new cells, but that's what cancer is. Cancer is your on-off switch is messed up, so you're constantly making new cells. And then I explained how that happens was because your on-off switch, so to speak, it's just a bunch of proteins, right? Proteins that attach to the chromosomes, proteins that attach to other things, right? So it's a bunch of little proteins working in concert. So if you mess one of those proteins up, you have a chance of messing up the whole system. And obviously, the bigger the mess up, the bigger chance you have of messing up the system and giving yourself cancer. Now, as you know, proteins are made by genes, right? A gene, as we already know from lab, that is a set of instructions on how to build a protein, right? So it's a long strand of DNA, which we're going to get into more in the next chapter, with A's and T's and C's and G's, and those letters say, all right, this is what you put together to build a protein. Now, when mitosis goes wrong, remember we learned in S phase of interphase, that's where the chromosome is duplicated, right? You did learn that. What I didn't tell you was that mistakes, maybe I did tell you. Anyway, mistakes happen. So it's not a perfect copy. So when you go through mitosis, it's supposed to be a perfect copy, but sometimes mistakes happen. So as long as you mess up one copy, and then you've got this new cell, then even that cell, as long as it's perfectly copied from now on, it's still going to be copied with that mistake because it already made the mistake, right? So you have to think, once you have one mistake, it's going to get worse and worse because you're going to keep adding mistakes and adding mistakes. It's a copy of a copy of a copy. So that's what happens when mitosis goes wrong, is you have, that's one way of getting mutations, which could give you cancer. Could give you other stuff too, but anyway, with that out of the way, let's talk about what happens when meiosis goes wrong, because that's what we just finished talking about is meiosis. What happens with errors in meiosis? Your book points out that it can result in genetic abnormalities, obviously, because if you mess up your chromosomes, you're going to have genetic abnormalities. They could be very small. They could be very large. So they could basically range from mild to severe to fatal. And there's necessarily not a reason to write that down. This is just an introduction to what we're about to talk about. I won't ask you that. That will not be a question. So knowing that meiosis, the purpose of meiosis is to first separate the homologous chromosomes and then separate the sister chromatids. Can anyone tell me, basically, forget the abnormalities, just tell me, knowing what meiosis is supposed to do, what would be the result if meiosis goes wrong? There's a simple answer, but the hard part is going to be not to overthink it. What is the simple answer? If something goes wrong in meiosis, what are you gonna be left with?
It won't reproduce.
It could. There's a possibility it won't reproduce, and we'll talk about that later. I mean, that's a lot of times that's what miscarriage comes from. There was an issue in meiosis, and because of that issue, and I don't want to give it away, but something goes wrong, and then the thing just doesn't make it. Yeah, fertilization happens, but the thing doesn't make it. So what is it that you're left with?
Abnormalities.
Yep, abnormalities.
Like a cleft lip could be something like that.
That's very specific and correct. Broaden it out. Just think about like cells that we've drawn. Think about all the different drawings you've seen of meiosis happening and what you're left with.
I mean, I've seen people born with one hand or extra finger, extra toes.
Yep, that's possible. But nobody? Just think about the drawings. Because you're right. I'm trying to be much more broad and not so specific. There you go. So the purpose of meiosis is to split up homologous chromosomes and sister chromatids so that you end up with 4 genetically unique cells, all haploid, right? You start diploid, which means you have both sets of chromosomes. And at the end, you should have 4 chromosomes that have half the amount of chromosomes. 4 cells that have half the amount of chromosomes. So if something goes wrong, then that just means you're going to have the wrong number of chromosomes. That's it. Everything else is due to that, right? All the stuff that Thornabar correctly mentioned happens because you have the wrong number of chromosomes. Of course, there's other reasons that stuff could happen too, but here we go. First of all, let's talk about how this happens. And the way you get this is something called non-disjunction. And that's just simply, again, meiosis. All we talked about was those chromosomes separating. So non-disjunction is when they're just not separating. Could be the homologous chromosomes not separating the first time, or it could be the sister chromatids not separating the second time. And it's in the name, right? Non-disjunction. That just sounds like what it would be. It's when the chromosomes do not separate. The next slide I'm going to show you is basically just a picture of what's on this board right now, the words that are on the board. I'll give you guys a second. I see notes being taken. If you're watching the video, and I haven't put out questions yet, well, so you guys are live, so obviously all you have to do is just email me. But if you're watching the video and I haven't posted questions yet, send me an email for attendance. Tell me what date it is. Like today, anyway, is October 15th. And today's keyword, I think last time it was cell phone. Today's keyword is dry erase marker. So send me an email. Say, hey, I'm writing for my attendance on October 15th. And the keyword is dry erase marker. And for those of you who are watching this video after the questions are posted, then I'm sorry. You just have to do the questions. But this is just me making sure that these people are who are here are actually paying attention. And it's okay if you're not. That's why this is being recorded and why I provide questions. So anyway, any questions about this slide before I show you the picture, the next slide, which is a picture of all this stuff? All right, like I said, it can happen to meiosis 1, it could happen to meiosis 2, meaning it could be the, it could be the homologous chromosomes that don't separate, or it could be the sister chromatids that don't separate. Either way, you're gonna be left with some mistakes. So if it happens in meiosis 1, you can see here that these two chromosomes on the left, I keep making that mistake today. These two cells on the left have one too many chromosomes. These two cells on the right have one too few chromosomes, right? So if that happens in meiosis 1, you're in a lot of trouble, right? Not a single one of your cells that you produce is gonna be normal. Now, if it happens in meiosis 2, right, at least you produced 2 normal cells during your first round, right? Then this thing messes up. So now you have these 2 that are messed up. So this one has 1 too many chromosomes. This one has 1 too few, but at least these 2 are normal. So I doubt I would ask this, but if I asked any questions, it would be for extra credit. And I might say, what is worse, meiosis 1 or non-disjunction in meiosis 1 or non-disjunction in meiosis 2? And the simple answer is non-disjunction in meiosis 1. Because again, look, all the cells are messed up if it happens in meiosis 1, which makes sense because you're splitting it and you're splitting it again. So if you mess up during the first split, then the rest of them are messed up. Now, when you do your study guide, it actually gets specific and asks questions. Like it might say, which one results in 4 cells with N+1 and N-1? Or which one results in 2 cells that are normal and 2 cells that aren't? And again, I probably won't do that, but if I do, it'll be extra credit. Any questions about this slide? All right. So keep in mind, this is just meiosis. So here we're just trying to produce gametes, right? This is mistakes in producing the gametes. Whether we're producing sperm or egg, that's what the mistake is. Now, let's talk about what happens from that. And the next slide I'm going to show you shows you a picture of it. And again, it's a lot of details that might be on your study guide, but I won't have it on the exam. And if I do, it'll be extra credit. But what we're looking at here in this next slide is going to be an abnormal gamete. So maybe a sperm, maybe an egg, whatever, fertilizing a normal gamete. You have to understand it's probably the chances of having an abnormal sperm meeting with an abnormal egg are probably pretty low. But anyway, so this is having one or the other. What happens is you get a zygote, and there's a word you need to remember, and that should remind you that when you mix a gamete and a gamete, you get a zygote. So that would definitely be a test question. When you mix a haploid gamete and a haploid gamete, you get a diploid zygote. Anyway, in this case, this particular case, not always, but in this particular case, This will make more sense when I show you the picture. The result is a zygote that has one extra chromosome. And because that is the one cell, right? The zygote is the very beginning, right? At one point in your life, all of you, you were one cell. When your mom and your dad, when those cells came together, that fused and made that one cell zygote. And ever since then, the way you grew was through mitosis. You copied the cells. You went from one cell to 2 to 4 to 8, 16, so on and so forth. So because that's the case, because we're using mitosis, then that mistake, that extra chromosome, that's getting copied every time. So that means every single cell in that person's body is going to have an extra chromosome, which is completely different than, like, let's say you're on vacation and, you know, it's Chernobyl and you get exposed to some really bad radiation in your arm or something. And now there was a mistake in mitosis and now you have extra chromosomes or not enough chromosomes and they keep duplicating. Well, generally speaking, unless it spreads, you know, the mistake is only in that one spot that was exposed. But here, because the mistake happened before you even created, that is just you for the rest of your life. If the organism survives, and that's a big if, it has an abnormal karyotype. And if you remember, a karyotype is just a picture that shows all the chromosomes. And it can have syndromes or disorders that are caused by abnormal number of genes. And we'll talk about that here in a second too. Here's a picture of what we're talking about. Here we have this abnormal egg. It has an extra chromosome with a normal sperm. And I'm thinking your book may have done this on purpose because one of the more common abnormalities we're going to talk about is Down syndrome. And the risks of a woman producing an egg with an extra chromosome go up after she turns, I think, 35. So maybe that's why they chose the egg instead of the sperm. That's a good independent work question for you because I don't know the answer. I know Down syndrome is, you know, the risks go up because of the mom being of a certain age. What about all these other syndromes? Also, Down syndrome is just one extra chromosome, number 21. What about all the other extra chromosomes? What if you have an extra chromosome, number 7 or 6 or 1? Any of the 23? What are those abnormalities called? Are they deadly? Are they caused by the male or the female more often? All those things you can think of. But anyway, abnormal egg meets with a normal sperm. Here we have an abnormal zygote because you can see here it has this extra chromosome. So what happens? It goes to mitosis and every single cell in this person's body is going to have that abnormal chromosome. Woo! So let's talk about some of these genetic malfunctions, these syndromes. And again, I've already mentioned this one because this is the big one, Down syndrome. It's also known as trisomy 21. That should be easy to remember. I might ask you about that on the exam. Tri should tell you, well, first of all, you should know that for each of your chromosomes, you only have 2 of them, right? You should only have a mom chromosome and a dad chromosome, right? You should only have 1 pair. So this whole tri thing means, oh, that's 3 instead of 2. So if you can remember tri and then 21, meaning it's on chromosome number 21, you should be good. Obviously, if you have 1 extra chromosome than you should have 46 chromosomes. So what's 46 plus 1? 47, right? Your book goes into detail about some Down syndrome, and we'll talk about it, but I won't ask you about this. It does affect 1 out of every 850 children. That's according to your book, and I don't remember what kind of book detail your book gets into. It's another independent work question if you want. Is that 1 in 850 children in the developed world, or is that in the United States, or is that worldwide? I don't know. It is the most common chromosome number abnormality, which is why we're talking about it, because, again, there's a bunch of other ones that you can look up for independent work, but this is the most common. And it is also the most common serious birth defect in the U.S., which leads me to believe, what about other countries? What are the other leading birth defects in other countries? Like I already said, the risk of this happening increases with the age of the mother. And anybody over the age of 35, we call that advanced maternal age. And because of that, they have prenatal screenings, and it's just a higher risk pregnancy when the mom is over 35. So again, none of these little bullet points right here are going to be on the exam. If anything, just know that trisomy 21, that's the condition, right? And that causes the syndrome of Down syndrome. So those things, as far as we're concerned, are synonymous. Down syndrome equals trisomy 21. Any questions about this slide? The next slide I'm going to show you gives you some characteristics of people with Down syndrome. I'm not going to ask you this on the exam either. They have characteristic facial features. I'm sure you guys have seen somebody with Down syndrome, and you can almost always tell. You look at them and say, that person has Down syndrome. They very often have a shorter lifespan, not always. I put a little star on there to remind you. That's a good independent work question. What is the average lifespan for somebody with Down syndrome? The average lifespan for humans, for the most part, has been increasing. But has that also been true for people with Down syndrome? Has their life expectancy also been increasing at the same rate? Also, and you can look this up, it has nothing to do with what I'm talking about, but I just remembered. If I remember correctly, I think for the first time ever, or the first time in a long time, maybe the average life expectancy of humans just went down a little because of stuff. I don't know. I'm not an epidemiologist, but I think I read that somewhere. Anyway, Down syndrome is not like a one-size-fits-all thing. There's varying degrees of developmental delays. Like some are more affected than others. Some may, well, I guess these two should go together. But some people with Down syndrome may live to middle age or beyond, and many of them are socially adept. This is all from your textbook, so I included it, but I'm not going to test you on any of this. But yeah, if you want, again, if you're interested in this, look it up for independent work. Look up the other syndromes. And also, this is obviously just scratching the surface of Down syndrome. What are some of the other stuff? Yes, characteristic facial features, but what are the characteristic facial features? And what other problems do they have? Anyway, moving forward, here's a normal karyotype. How do I know it's normal? Because look at right there. Well, first of all, let's look at all of them. Every single chromosome is there, from number 1 all the way to the sex chromosomes. Every single chromosome only has one of each, right? So there's a pair for each, including specifically the one we're talking about here, 21. This particular one only has a pair of chromosome 21. This next one, trisomy 21, right? This cute little baby has an extra chromosome on chromosome 21. And I have a hypothesis, and I haven't looked this up. Someone may have already looked this up. You can look this up for independent work too. We know that genes are on chromosomes, right? That's what chromosomes are. It's DNA that has genes on it. And here we go. Here's a structure function thing. I can tell you right now by looking at chromosome number 1 versus chromosome 22, for example. Look at the size of them. I'm willing to bet that chromosome number 1 has many, many, many more genes than chromosome number 22. Does that make sense? I could be wrong, because as you're going to learn in the next chapter, there's things called introns and extrons and this so-called junk DNA. But anyway, I would venture to guess that there's many more genes on chromosome 1 than 22. So what I'm thinking is, possibly, my hypothesis is maybe the reason that trisomy 21 is so common is because it's such a small chromosome, which has such a small number of genes, that it makes not as big of a difference. So it's more survivable. Because I know a lot of these, and I don't know specifically which, but I know a lot of these, if you have an extra chromosome, you just don't make it past pregnancy. So maybe that's what it is. And you can look that up if you want, because I really don't know. Maybe if you have an extra chromosome number 1, that's such a big difference because it's such a huge chromosome. Maybe that's just, a non-viable zygote. Just a hypothesis for me. I'm sure somebody already knows. I'm sure that study's been done. But anyway, any questions about that? All right. Moving right along. Abnormalities of sex chromosomes. So we're just going to skip all the other ones. That's why I said it's a great independent work topic if you want. What about all those other 22 chromosomes, right? We've only talked about 21. What about 1 through 19, or 1 through 20, and then also 22? But anyway, let's talk about sex chromosomes now. Obviously, non-disjunction can affect more than chromosomes 1 through 22. It can also affect your sex chromosomes. But that seems to upset things a lot less. So it's not usually as big of a deal as something like Down syndrome or whatever syndrome might happen when you have an extra number 1 chromosome, for example. So there's not much to say. I probably won't ask you anything about this on the exam. Just know that non-disjunction can also happen in sex chromosomes. Your book gives a whole chart here. So this is what happens when you have an extra X chromosome, right? You're still a male because you have a Y chromosome, but you have that extra female chromosome. And it gives you the frequency. So 1 in every 2,000 people have it. 1 in every 2,000 people have an extra Y chromosome. 1 in every 1,000 people or females have nothing, an extra X chromosome. Some females only have 1 X chromosome. That's what that little O stands for. So none of that will be on the exam. You can look it up, though, because it doesn't tell you much of what happens. You got the symptoms down here a little bit, but I'm sure that's just scratching the surface. What else can happen? And how many of you are criminal justice majors? Well, we got 1 in person. I don't know if any of you are online are. And this is something I've read. I'm not sure this is true because I don't have the source in front of me. So it could have been some garbage source. But there is something called a super male. I've heard it called this XYY, right? So normal male is XY. Y being that chromosome that makes you a male, right? So some males have an extra Y chromosome. And I think and you should look this up to be sure, but I think the people with the extra Y chromosome are a little bit extra male, meaning they have like extra testosterone, and with that, what comes along with that, which would be extra body hair, extra aggression. And I think I read somewhere that there was a study where they looked at the general population. We know that 1 in every 2,000 people have that, right, in the population. But they looked at the population of people in prison for violent crimes or something like that, and the percentage was higher. So you can look that up if you want. I thought that was interesting. Like the fact that you have an extra chromosome makes you more aggressive, more prone to commit violent crimes. It's a very interesting thing. You could really go down a wormhole, a rabbit hole on that one. Because then you get into ethics. Like, all right, so do we prejudge somebody because they're genetically predisposed to be violent? Should we take precautions with those people? Or should we give them, treat them like everybody else? I have my opinions, but I won't go into that. Anyway, that's it. Every chapter ends with this right here, the evolution connection. Yeah, there's questions. Let me go through this very quickly. Usually I skip it. But this is on your study guide, and it might be on your exam. Basically, it's like the question is asking, why do we even have sexual reproduction? Because it used to be it was all asexual. Sexual reproduction is a result of evolution. So the question is, why is there an advantage? And you do need to know this. Well, first of all, let's talk about asexual reproduction. If you are an organism like a bacteria and you're living in an environment that's not changing and you're just really great, you're really fit for that environment, right? You take advantage of the heat, the pH, all the food that you're getting is perfect for you and it doesn't change. So since you're so perfect in that non-changing environment, evolutionarily, it makes sense for you to make a perfect copy of yourself. Mitosis, asexual reproduction. However, if you're in a situation where, that's what I just talked about here, right? But if you're in a situation where things are not so perfect, right? Where things are constantly changing, then it makes sense to have variation. That way, all my offspring are going to be slightly different. And that way, when the climate slightly changes, one of them will have a better chance of making it than the rest, so to speak. So anyway, that is a question on your study guide. Make sure you study it. Let me know if you have questions. I will see you all on Monday or during office hours, which I'm still giving extra points for attending.